Hi everyone, so this will serve as your pre-recorded video pertaining to the lesson shapes or lesson 4 shapes and sizes of bacteria. So as a review, I will be asking you with regards to classification of microorganism next week or maybe tomorrow if we will be having a face-to-face. So this will be the topic outline. When I say topic outline, this will serve as your standard, your basis on what we will be discussing for today. So we will be discussing the structural and characteristic of bacteria, the features pertaining on salient or those features that bacteria have, the morphology of bacterial cell, or when we say morphology, it is a formation of the bacterial cell. We will also discuss the sizes and the shape, and also the different shapes of the bacteria. And aside from that, we will also discuss the movement of the bacteria. What are those bacteria that is motile and what are those bacteria that is non-motile. So considering, let's move forward on the very first term that we will be encountering, and that would be bacteria. So as we all know, when we say bacteria, it is a prokaryotic cell. When we say prokaryotic cell, it has only one cell, and that is called unicellular microorganism. Bacteria is also can be found everywhere. That's the reason why it is most abundant. When we say most abundant, each areas you can found or you can get bacteria. And of course, not only that, aside from that areas, those extreme areas, we can also found or find bacteria. What are those extreme areas that we have? We have hot springs, snow, deep ocean, or those difficult areas that a normal organism cannot live, bacteria can live. Okay, so that is the definition of bacteria. So now moving forward, this illustration will show you the microscopy view of bacteria. And aside from that, this is also the representation of the parts of prokaryotic cell, because as we all know, bacteria is a prokaryotic cell. So aside from that, we will be unlocking also the characteristics of it, the definition. But for this slide, we've already done this in discussing what are those, we already done this in your anatomy, so I will not be explaining it furthermore because I know you already know this one because you passed your anatomy and physiology. The only term that you are very not familiar is the endospore. When we say endospore, it is in the outer layer of the prokaryotic cell, and it serves as your protection against what? Environmental condition such as heat or drought. So that is the endosperm. So moving forward, let's discuss what are those features or important features pertaining to bacteria. Number one, it is unicellular, meaning to say it has only one single cell that makes bacteria as prokaryotic cell. Features again, bacteria can be presented in different shape and sizes. They can also be presented in arrangement by the sizes. Bacteria also lack in nucleus and membrane-bound cell organelles, but bacteria can be perform all the needs in nucleus in the serve of cytoplasm. While because in the bacterial DNA, you can be found it in cytoplasm and not packaged to form chromatin as in eukaryotic cell. In eukaryotic cell, you can find the DNA or the process in DNA in the nucleus. But here in bacteria or in prokaryotic, you can find it in the cytoplasm. Bacteria also is 10 times smaller than the human cell. As we all know, human cell is very, very small. But to be exact, we have 0.2 nanometer to 10 to 50 diameter. But here in the constant sizes of bacteria, we have 10 to 6 diameter. So it's very, if we compare it, it's very small if we compare it to human cells. So moving forward, we have also the covering layer of the bacterial cell is called cell wall. As we all know, going back to your topic in human anatomy, when we say cell wall, it confers rigidity. When we say rigidity, it is not changes or not changeable of what? Shapes or sizes. So meaning to say bacteria has its own definite shape and constant sizes. So the bacteria also is made up, or the cell wall of the bacteria is made up of peptoglycan or murein. When we say peptoglycan, it is a kind or a combination of amino acid, and when we say amino acid, it is a building blocks of protein. Meaning to say bacterial cell consists of protein synthesized amino acid. So The features also of the salient is in different shapes, and different shapes by different bacteria species. And also it contains what? Cilia and flagella. It is very important for you to know the for you to know the importance of having the cilia and flagella, because these are the two parts of prokaryotic cell that is much more on the movement of the bacteria. And bacteria also can be identified into photoautotrophs and chemoautotrophs. So when we say photoautotrophs, you know already this one because I've already discussed this when we say photoautotrophs, it is the organism that can produce by their own food. So meaning to say they can produce their own food from the process of photosynthesis, from the energy coming from the sun. Next, we have the chemoautotrophs. When we say chemoautotrophs, these are the bacteria that can live, or that these are the organism that can live with the presence of chemical reaction or chemical bonding. What particular chemical reaction is being processed? We have the nitrogen fixation, one of the example, and what else? Evaporation, that is one of the example on how the bacterial cell can get food throughout the processes of chemicals. Okay, then let's move forward on the formation of the bacterial cell. There are, I think, much more two explanation or two discussion pertaining to morphology or the formation. So the morphology or the shape of bacterial cell can be distinguished by its property. So meaning to say, not only the shapes or the illustration itself, but we can determine the formation of the bacteria by its property. Okay, by its property, or by the characteristics of the property of particular species of the bacteria. Next, we have morphology of bacterial cell. So this entails also not only with the shapes, but it also entails how pathogenicity can occur with the bacteria. So as we all know, one good types of pathogen is bacteria. Okay? So the important factors of bacterial cell can also adapt in evolution. So there are many features like motility, mode of nutrition are affected by the shape of bacteria. So meaning to say, in the morphology or in the shape of bacterial cell, we cannot say that we will just be focusing on the shape itself, but hence we will be focusing on also how motile the bacteria is. When we say motile, how does the bacteria move, or what particular movement that it's occur. Then we also have including in the morphology of the cell, the peptoglycan. As I've said, peptoglycan is made up of what? Our cell wall is made up of peptoglycan or marine, which is polymer of sugars or long chain sugars that consists of what? Peptide chain called N-acetylglucosamine and N-acetylmuramic acid. That's why it is not only sugars, but also it can be defined into what? Building blocks of protein, which is the amino acid. So it's very responsible for the varied morphology and the shape of the bacteria. Next, we also have the sizes of bacteria. For the sizes of bacteria, the constant is 2 micrometers with a diameter of 0.5. So that's why if we compare it to the human cell, it is very small or smaller than the human cell. So bacteria also can be displayed in wide variety of shape and about 10 times smaller sizes of eukaryotic cells. So these are the particular sizes of its microorganism. Okay, so let's move forward on the different shapes of bacteria. So as you can see, we have cocci, bacilli, the budding, and the others. Okay, so I want you to draw each shapes and sizes, and I will be checking it tomorrow. Okay, so for the shapes of bacteria, we will be starting to identify the spherical cocci. Then next, we have the rod-shaped bacilli and the spiral shape and the others. So as I've said, there are different shapes of bacteria, starting with the spherical, which is cocci, rod shape, which is bacilli, and bacilli spiral bacteria, and comma shape, which is Vibrio. So now let's start on identifying the bacteria that is possesses two or arranged into spherical. So for the arrangement of cocci, if there is only one bacteria, it possesses only what? Coccus. So if it is diplo or diplococci, it is consists of two bacteria, two formation of bacteria. When we say staphylococci, it consists of um 10 bacteria. So meaning to say, when we say spherical, it is from a single to multiple group from 2, 4, or 8. Okay? So cocci bacteria can be also round or oval. So as you can see here, it is elongated and it is like a bin shape. So the cell wall coccus shaped bacteria may be gram-positive with thick peptoglycan layer or gram-negative with thin peptoglycal cell wall. So I want you to familiarize that one because this can explain, or this particular process is the gram staining, right? We've already talked about it last topic. From when we say gram staining, it is applicable to bacteria. So in cocci or in coccus, gram staining bacteria, gram positive with thick peptoglycan layer and gram negative with thin peptoglycan cell wall. So that is in the cocci. The cells here in coccus do not separate after cell division. So if we literally compare it to human cell, the cell in human can be performed in cell division. So thus the plane of the cell division changes by its shapes of the bacteria. So according to arrangement itself, there are different types of arrangements of cell pertaining to cocci. It will start with, of course, the single one, and that is monococcus. When we say monococcus, it is only consists or exists in single spherical cell, while in diplococcus, from the word itself di, it consists or it's arranged by pair after the cell division. So one good example of diplococcus is diplococcus bacteria that is gram negative and gram positive. Gram negative Neisseria moraxelia cataralis, and gram positive Streptococcus pneumonia enterococcus. So that is the example of diplococcus. So aside from that, we also have Streptococcus. Here, the cocci are joined in the plane and arranged in chain pattern. So these are non-motile, meaning to say it is not considered in the movement, or it is not much more focused on the movement of the bacteria, and it is aerobic presence of air, and has a gram positive bacteria that cause of many diseases. So streptococcus is much more digging deep with the gram positive bacteria, and when we say gram positive, it possesses what? Many diseases. And as you can see here, these are the examples of streptococcus. The next one, we have the tetrads. Tetrads is cocci that is consists of four formation. These are arranged in group of 4 of cell, and it is usually or the cell division will usually occurs in two differential plane, because you will be divided into two. So the appearance of it, or the illustration of it, is in group, okay, in group of 2, but the formation of it is in 4. Okay, so example of tetrads are bacteria like Micrococcus and Pediococcus. So we also have the Staphylococcus. When we say Staphylococcus, these are arranged in a regular cluster, so which looks like a grapes. Staphy is much more on 10 formation of coccus. This is due to the cell division in 3 planes, so meaning to say it is divided into 3 planes. In tetrads, it is divided into 2, so that makes 4 group, but here it divides into 3 planes. One good example of Staphylococcus are Staphylococcus aureus and Staphylococcus hemolyticus. So that would be the examples of Staphylococcus. Next, we have Sarinae. When we say Sarinae bacteria, these are anaerobic or has no presence of air. Anaerobic gram-positive bacteria, and they may occur in or can be found in the large intestine or the skin. So one good example of SARNA are the terms that is being said in the slides. And when we say SARNA, it is much more can be found in your intestinal tract or in your stomach. So now let's move forward on the rod shape. As you can see, if we trying to compare the both shape from the cocci to bacilli, as you can see in cocci it's much more spherical, but here it is elongated or rod shape. So Cucubacillus is only one, while bacilli it is much more larger to cucus. It is much more like not elongated, but it will change into shape, and so on so forth. So these are the rod shaped bacteria. They also occur in single or multiple cells. So meaning to say, not only cocci is possesses on single cell, but here in bacillus it also possesses with single, but not only single but also in multiple cell, and it is usually attached after the cell division. So the bacilliform bacteria are found in many different taxonomic group of bacteria. So it can be gram positive or gram negative. So these are, I think, the feature of bacillus. So in bacillus, it is much more focused on the gram positive, but not in a sense that it will always gram positive, but hence it will also be gram negative. So meaning to say, it's either gram positive or gram negative. They are obligate aerobe or facultative anaerobe bacteria on anaerobic bacteria. So meaning to say, since it is possesses of gram negative and gram positive, it also possesses aerobic and anaerobic. A presence of air, there are bacilli that has a presence of air. There are bacillus that is not, or no presence of air. So they can form also in endosperm or in the outer layer of the cell or in the bacterial cell, and they can live in years. They can survive also in extreme heat, like for example 420 degrees Celsius, and it is the most abundant bacteria that found everywhere. So in cocci, cocci is usually can be found in some of the areas, but the most abundant bacteria shape is the bacillus. So it is mostly non-parasitic. It is a free-living species, and these are so meaning to say, there are two parasitic and pathogenic bacillus species, and there are causing by anthrax and bacillus cereus, which causes food poisoning. So meaning to say, not only in the areas in the environment, so even in the food you can get bacilli or bacillus shaped bacteria. So these are the arrangement based on the bacilli. When we say single, it is the bacillus. So if there is no attachment of other cell, it is only single rod, it is bacillus. If it is double or two rod that is attached to one another and it begins after the cell division, it is diplobacilli. And I want you to familiarize also the examples, ha. Then we have streptobacilli. It can be done after or during the cell division, and it arranged by one plain bacilli. Okay, and it also contains a gram negative and aerobic or facultative anaerobic. So it possesses into two, anaerobic and aerobic, while the cocobacilli, these are short compared to other bacilli. It is usually an oval in shape. Pag cocobacilli, it consists also with one single cell. Okay, they also appear like a coccus. It's like a coccus. It is unelongated. It is smaller than bacillus. Then lastly, we have palisades. So these are the bacilli after cell division, and after the cell division it will bend, and therefore it will arrange in palisade and fence. When we say palisade or palisade, it is much more on, or the illustration of it is fence like, yung parang sa gate nyo, parang fence like structure, and that is a good example or illustration of palisades. Okay, so now let's move forward to spiral bacteria. Here in spiral bacteria, there are four. We have the Crooker's form. Pag sinabi nating spiral, it's like a DNA illustration that it can bend thoroughly because it's a spiral. So the arrangement of spiral can be consist on the spirillum. Number one, Spirilium consists of gram-negative, and it is rigid, meaning to say not changeable in sizes, and this bacteria is having a flagella, or the part of prokaryotic cell that can move, or why the prokaryotic cell can be moved because of the presence of bacteria. Then next we have the spirochete. When we say spirochete, it is much more pertaining on the spirality or spiral of the formation or the shape. It is usually flexible, meaning to say you can flex, you can change the shape of it, and it is also have a, if the spirillum has an external flagella, here the spirochete has an internal periplasmic flagellum or flagella. It is much more on the movement. It's much more focused also with the movement. So these are the pathogenic species that cause various serious diseases. So that is pertaining to spirochete. One good examples are Leptospira, Treponema, and Pallidum. So these are the examples. So I want you to familiarize all the examples, ha? Okay. Next, we have the comma shape. Comma shape Vibrio, or this is usually can be found in the curve and appear like a coma. So these are mostly gram-negative bacteria, and it is known as a foodborne diseases. And usually Vibrio can be facultative and aerobes, or has no presence of air, but it consists of two chromosomes which replicates independently, and that makes it a comma shape. So the main four shapes of the comma Vibrio are the filamentous, or when we say filamentous, it has a long filamentous structure. Okay? Parang it's like a sperm style shape. Then pag sinabi nating star shape, it has a resemblance of star. And when we say rectangular, these are the Vibrio that can form into box or rectangular shape. And when we say pleomorphic, it is a ability to change their shape and sizes due to the various external factors like pressure or environmental stress. So filamentous, it is like a long chain filamentous structure that is actually been basically can be compared into a sperm or into a illustration of a sperm. When we say star shape, very simple. It has a resemblance of the star. And when we say rectangular, these are the Vibrio that can be possesses, or the formation of it is can be defined into boxes or rectangular. And lastly, we have phylomorphic. It is much more particularly on the ability of the shape in Vibrio that is changing. Okay? So again, I want you to familiarize the example so that it is in there, so I will not be reading it. Okay? So aside from these size four shapes, this also the other main shapes of bacterial cells that possesses various different morphology, namely appendage. So there are this appendage is also known as a budding bacteria. They can be non-motile or motile. Pag sinabi nating non-motile or motile with flagella, they can be possesses into movement, or they cannot be possesses into move, or they cannot move. So these are the examples. We have trichome, or these are the chain of vegetative, and this is often covered with the slimy sheet as cyanobacteria. So this is trichome, and it is usually a chain that covers a slimy sheet. When we say slimy sheet, it is possesses also a water and plasma. Then we also have the lobe. When we say lobe, these are the structure that mostly can be found mostly can be found in not in the cold area, but hence it can be found in the hot and volcanic spring. So they are usually flagellated, or they can move faster or move freely, but the shape of it is irregular. So they are also, one good example of it is acidophiles and thermophiles, because in our previous lesson we already know what are the acidophiles and thermophiles, diba? So these are particularly a bacteria that can be found into hot spring, or those areas that is very hot or warm. Okay, so now let's move also with the another shapes in bacteria, and that is fusiform or falciform. Fusiform or falciform is a spindle shape. Spindle, when we say spindle, it is like most specifically is like a spiral also that bulge in the middle and in the end is tapered. Okay? So what are those example of fusiform? We have Fusobacterium and Necrophorum. So fusiform is a spindle shape that is possesses in the morphology of the bacterial cell. Remember, everyone, you have to know the shape of the bacteria and the arrangement of the bacteria. Next, we have the stalk. Stalk develops one end of bacteria due to asymmetrical cell division. Okay, when we say asymmetrical cell division, it is not only possesses on the nuclear division, but it also undergoes with cytokinesis. Okay, and lastly, we have the sheet. The sheet is covered from the word itself, meaning to say it's covered with the cell wall or with the sheet that performs what? Rigidity. So that is the shapes of bacteria. And now moving forward, we have the motile and non-motile. So the organism motility is refers to its capacity for self-propelled movement while utilizing metabolic energy. So meaning to say motility or having motility is propelled with movement. Okay? It's propelled with movement, and these are the bacteria that possesses what? Movement. And when we say non-motile bacteria, these are a type of bacteria that don't have capabilities or physical capabilities to move. So kaya nga tinawag natin siyang non-motile because it has no presence of movement. These are the bacteria that only develop along the stub line or cultivated stub. So it is along the line but not not possesses into movement. But not non-motility or non-motile bacteria can spread throughout, but not that, but they are not, cannot move. Okay? So that is the motility and non-motility of the bacteria. So these are the examples of the bacteria that possesses in motile, and these are the examples of the bacteria in non-motile. Okay? So I want you to familiarize all of that. So these are the results of the motility test. So this is much more focused on the bacterial cells. So I think that's the end of the shapes of bacteria. So if you have questions and clarifications in my pre-recorded video, you can ask me directly, or you can ask me in GC, or even in personal. Okay, or you can ask me personally. So your assignment is to research at least 15 pathogens or bacteria that can cause disease and follow the format. And dito ako magbigay ng instruction, ha? And write it in a short bond paper. Okay? Follow the format. So I think that's the end of our lesson 4. So if you have questions, clarifications, you can directly ask me by PM in Messenger, or you can directly ask me in personally. Okay? Thank you so much. Bye.